Look upon the ash. Long before our recorded history, a cataclysm wiped the slate clean. Yet, from the ruins of this forgotten epoch, a divergent genetic lineage quietly emerged, one that would defy the very laws of terrestrial evolution. In the sterile quiet of a modern laboratory, the truth of our origins is spun at thousands of revolutions per minute. Here, blood is separated into its elemental components, exposing a silent, molecular rebellion. For 85% of humanity, the red blood cells carry the Rhesus antigen, a protein inherited directly from our primate ancestors. It is a biological signature, a chemical thread linking us to the great apes of the Miocene epoch. But for the remaining 15%, this ancient primate signature is entirely absent. There is no mutation, no variation, simply a profound, empty void where the antigen should be. A genetic deletion of absolute, chilling precision. This genetic anomaly is not evenly distributed across our world. In the isolated valleys of the Pyrenees, the Basque people harbor the highest concentration of Rh-negative blood on Earth. Their language, like their blood, has no known relatives. This is not a gradual adaptation. In evolutionary biology, a complete gene deletion is a rare and radical event. The Rhd gene did not slowly degrade over millennia, it was cleanly excised, leaving a genetic scar that defies standard Darwinian models of gradualism. This deletion triggers a terrifying biological conflict. When an Rh-negative mother carries an Rh-positive fetus, her body does not recognize the child as her own. Instead, her immune system identifies the baby as a foreign invader, launching a relentless, lethal chemical assault. This is hemolytic disease. It is an evolutionary impossibility. Nowhere else in the entire natural kingdom does a mother's body naturally synthesize antibodies to destroy her own developing offspring. It violates the most fundamental law of survival, the preservation of the species. Why would nature select a trait that actively threatens reproduction? To find the source, we must journey back to the Neolithic transition, 10,000 years ago. As humanity abandoned foraging for agriculture, this mysterious blood type suddenly surged in frequency, coinciding with a massive leap in human culture and technology. In deep, subterranean chambers, scientists extract ancient DNA from the petrified bones of our ancestors. They seek the exact moment this genetic divergence occurred, drilling into the past with microscopic precision. Some researchers point to the Cro-Magnon people, highly advanced, robust hominids who suddenly entered Europe with artistic capabilities, complex tools, and larger brain capacities than the Neanderthals they replaced. Was the Rh-negative factor their defining genetic signature? How did this fragile genetic anomaly survive? In a world of dominant Rh-positive genes, the recessive Rh-negative trait should have been rapidly diluted and driven to extinction. Yet, it persisted, preserved in isolated pockets like an intentional genetic reservoir. Those who carry this blood often report strange, shared physiological anomalies, lower average body temperatures, unusually low blood pressure, a high occurrence of green or hazel eyes, and even a rare extravertebra in the lower spine. More elusive are the psychological reports. Across global surveys, Rh-negative individuals consistently report heightened sensory awareness, an unexplained sensitivity to electromagnetic fields, and vivid, recurring dreams of ancient, cataclysmic events. These anomalies have fueled alternative historical theories that bypass conventional evolutionary science altogether. They suggest that the Rh-negative blood type is not a natural mutation, but the result of a deliberate, prehistoric genetic modification. In the dusty ruins of Mesopotamia, clay tablets written in cuneiform tell of the Anunnaki, beings who descended from the heavens and modified the native hominids of Earth to create a specialized lineage. Could this ancient myth be a primitive description of genetic engineering? The texts describe the mixing of the divine life force with the dust of the earth. To ancient scribes, blood was the soul, the carrier of identity. A deliberate alteration of the blood would forever separate this new lineage from the rest of the natural world. Throughout history, royal dynasties have obsessed over the purity of their bloodlines, practicing extreme endogamy to prevent dilution. They called it the royal blood, a sacred inheritance that granted them the divine right to rule over the common populace. This obsession with blue blood may have a literal, chemical origin. While human blood is iron-based, some marine organisms use copper to transport oxygen, turning their blood a striking blue. Did an ancient, non-human lineage once possess a copper-based biology? The localized concentration of Rh-negative blood heavily challenges the simplistic out of Africa a model of human migration. If we all emerged from a single, homogeneous population, why does this radical genetic deletion cluster so intensely in specific European and North African enclaves? To solve this, geneticists have turned to our extinct cousins. By sequencing the genomes of Neanderthals and Denisovans, they hope to find the origin of the Rhd deletion, searching the deepest branches of the hominid family tree. The results were shocking. Neanderthals were overwhelmingly Rh positive. The deletion was not inherited from them. The mystery only deepened if the Rhd gene was intact in both our primate ancestors and our closest hominid cousins. Where did this deletion come from? It appeared suddenly, a massive genetic leap that coincided with the rapid expansion of the human neocortex. It is as if two distinct evolutionary trajectories were forcefully merged into a single species, creating a walking biological paradox. If this was a deliberate modification, what was the mechanism? The locus of the anomaly lies on chromosome 1, the largest human chromosome. Here, the Rhd gene is flanked by two highly identical DNA segments. This specific structure makes the gene highly unstable, prime for a targeted deletion event. When the deletion occurs, the body's immunological memory is altered forever. Once an Rh-negative mother is exposed to Rh-positive blood, her immune system creates permanent antibodies, a biological defense mechanism that never forgets and never forgives. In the mid-20th century, modern medicine developed RhoGAM, a specialized antibody treatment that masks the fetal Rh-positive cells from the mother's immune system, acting as a synthetic shield to prevent the natural rejection of the child. But before this medical intervention, how did Rh-negative lineages survive? In the ancient world, infant mortality among these mixed couples would have been devastatingly high, naturally suppressing the gene. Yet, somehow, the lineage was fiercely protected and maintained. Some scientists hypothesize that the Rh-negative mutation offers a hidden, powerful evolutionary advantage. Studies suggest that Rh-negative individuals possess a unique resistance to certain widespread parasites, such as Toxoplasma gondii, which can alter human behavior and reaction times. We find other isolated pockets of this blood type among the Berbers of North Africa and the Bedouins of the Sinai. These nomadic tribes, living in harsh, unforgiving environments, have kept their genetic pools isolated for thousands of years, preserving this ancient mystery. This geographic distribution has led some alternative historians to suggest a connection to lost civilizations. Could the Pyrenees, North Africa, and the Celtic fringes be the landing sites of refugees fleeing a sunken Atlantic continent? Others propose a cosmic origin. Could intense bursts of cosmic radiation from a nearby supernova have triggered a sudden, localized mutation in our ancestors, rewriting their genetic code overnight? The molecular structure of the Rhd gene remains a battleground of theories. Its complete absence in Rh-negative individuals is not a random mutation of single nucleotides, but a clean, structural deletion of the entire genetic sequence. Is it a design feature? Deep within secret archives, ancient codices, and forbidden genealogies are kept under lock and key. These texts, hidden from the public eye, track the movement of specific bloodlines through the rise and fall of empires. As we look to the future of genetic engineering, we must ask, are we simply rediscovering tools that were used on us eons ago? The Rh-negative anomaly stands as a silent witness to a chapter of our history that has been systematically erased. Today, global blood banks and government databases track blood types with unprecedented precision. The rare Rh-negative blood is highly sought after, catalogued like a precious commodity of unknown origin. Are we one single, homogeneous human family, or are we a hybrid species carrying the legacy of two distinct creators? The biological conflict in the womb suggests a deep, unresolved division in our very code. Until we unlock the secrets of chromosome 1, the Rh-negative anomaly will remain an evolutionary ghost, a silent, molecular testament to a forgotten origin story written in the very fluid of life.